Hi, my name is Marie Paceni. My article is entitled Deletion of P2X7 Attenuates Hyperoxia-Induced Acute Lung Injury via Inflammasome Suppression. It is written by Gallam et al. In the American Journal of Physiology, written on March 15th, 2016. So, people with pulmonary lung disease often require greater concentration of oxygen because in some way they are lacking. But at the same time, this greater concentration of oxygen can be detrimental to one's health. So, a greater partial pressure of oxygen within the lungs is known as hyperoxia. So, this greater partial pressure actually increases the reactive oxidase species, or also known as ROSs. So, ROSs are radicals that can lead to the damage of tissues and organs. At the same time, it can also increase inflammation, which is what my paper specifically will be focusing on. At the same time, these two can lead to acute lung injury, which could ultimately result in death. So my paper focuses on the gene P2X7. So P2X7 is an ATP-gated receptor. So ATP has to bind to it in order for it to open. It is present in macrophages, microglia, uterine and endometrium tissue, and also the retina. So what happens is P2X7 will bind to an inflammasome, as you will see in the next slide, and the binding to the inflammasome will induce an inflammatory response. So an inflammasome is made up of non-ligand receptor proteins, also known as NLRP1s, and they come together to form a multimolecular complex. So once again, P2X7 will activate this inflammasome, and the activation of the inflammasome will cause it to bind to procaspase enzymes. Once these procaspase enzymes are activated, they will turn into activated caspase enzymes, which will then cause other proteins to promote the maturation of cytokines such as interleukin-1 beta, interleukin-6, and also tumor necrosis factor alpha. So for their first experiment, they took 10 mice. 5 had a knockout for P2X7 and 5 had the wild type. So they put these 10 mice, which were 6 weeks old, and they placed them in a closed chamber. They then exposed them to 100% oxygen for a day, 2 days, and then 3 days. So remember, 100% oxygen simulates an hyperoxia environment. And then controls you will see in the later experiments were just exposed to regular room air, so just the normal concentration of oxygen. So as you can see, the wild type are the circles and the rectangles are the P2X7 knockouts, meaning that the P2X7 is no longer functioning properly. And when you look at this chart, you can see that the mice that had the P2X7 knockout had a greater rate of survival than the mice with the wild type of the P2X7. So the mice lived longer when their P2X7 protein was no longer functioning properly. So for their second experiment, what they did was they anesthetized the mice. Then they surgically exposed the trachea. This was done in order to collect the BALF. So the BALF is known as the bronchoalveolar lavage. So it basically just contains all the cells and fluids that would be within the bronchioles. Then what they did was they performed a BCA protein estimation assay to figure out the amount of proteins that were present within the BALF, so within each trachea of each mouse. So when they did this, they found that there was significantly more proteins within the BALF of mice that had a wild type P2X7 versus those that had the knockout of the P2X7. What you can also notice is that this only happens in hyperoxia. So if you look at the other two columns with the just room air, you don't see that difference between the wild type and the knockout. So because there's a greater in a greater concentration of proteins within the BLF collected, that means that there was a greater immune response within the wild type versus the knockout. So for their third experiment, they did the same procedure. So they anesthetized the mice, they surgically exposed the trachea, and they collected the BALF. But this time they took the BALF and they centrifuged it, took out the supernatant, and plated it so that they could see the amount of cytokines present within the BALF in knockout mice versus the wild type. Right, so now they want to see how P2X7 affects inflammation. So once again, they took the BLF samples and they quantified the amount of cytokines present within the BLF in wild type mice and also the mice that had the P2X7 knockout. So when they quantified these, they saw that then when the mice were exposed to a hyperoxia environment, that the wild type had significantly more IL beta, IL-6, and tumor necrosis factor cytokines versus the mice that had the P2X7 knockout. You can also see that when the mice were exposed to air that was just room air, normal concentration of oxygen, you did not see the same results. So the wild type and the P2X7 knockout had similar amounts of cytokines. So what can we conclude from this? We can conclude that P2X7 increases lethality with exposure to hyperoxia. We saw this in the first experiment. So the mice with the wild type P2X7 died earlier than the mice that had the knockout of the P2X7. We also saw that a wild type P2X7 causes a greater immune response versus mice that had the knockout P2X7 in a hyperoxia environment. Lastly, we saw that there was an increase of cytokines, interleukin-6, interleukin beta, and tumor necrosis factor when the wild type was present inside mice versus the knockout in a hyperoxia environment. So the researchers went on to do things such as provide antagonists for P2X7 to show how that would affect inflammation. They also looked at the amount of white blood cells in the region when P2X7 was functioning properly. And they found a stronger connection that P2X7 does cause inflammation, and it actually could be increasing acute lung injury. So my first question is inflammation an innate or adaptive response? And then my second question is, what kind of white blood cells would you expect to see in an inflammatory response? So this is part of the later experiments that they did that I did not include in my presentation. These are just my citations. Thank you for watching and listening.